Okay, I'm back again. We survived anatomy. We survived microbiology. So now it's time for anatomy. Am I right, guys? Or am I right? Let me just check that this live is working. Okay, so we are live. Time to share my screen. You guys, this is the first time I've streamed in the daytime, so you guys can see here's my bed. Here's my entertainment center. Down here's all my little games that I like to play. So let's share my screen. Dang, this won't even work already. How about this? All right, so this has already given me some backlash. Okay. All right, so we're here. So we're gonna do week 1 today. So let's go to our week 1 slots. So describe the major functions and structures of the respiratory system, contrast to conducting and respiratory zones. So conducting and the difference between a conducting and respiratory zone is that in the conducting zone is in the respiratory zone, there is gas exchange. So everywhere else that there is no gas exchange, including bronchioles, terminal bronchioles, our trachea is all conducting zone. Respiratory zones are respiratory bronchioles and alveoli, alveolar duct, alveolar pore. All right, so describe the 4 respiratory processes. Ventilation. Ventilation is just getting air in and out. External respiration is getting external air to exchange it into our blood. So I think of external environment. So air from oxygen from the external environment is getting breathed in. Internal respiration, I think of the internal environment. So that's where I think of capillaries and tissues. And cellular respiration. I don't know about cellular respiration. We haven't done that yet. Define pulmonary ventilation, inspiration, expiration, and relative values for atmospheric pressure, intrapulmonary pressure, intrapleural pressure, and transpulmonary pressure. Okay. I don't remember transpulmonary pressure, so that's good that we're going over it. Atmospheric pressure is just the pressure that we feel in the atmosphere, all the air that's above us and all the air that's next to us pressing against us. Intrapulmonary pressure is what's the pressure inside the lungs, and intrapleural pressure is the pressure inside of the pleural cavity. This was kind of hard for me to pick up at first just because I kept on thinking that there would be the same thing. But I have to remember that the lungs is kind of like the functional part, and the pleural cavity is still part of the lungs, or what we think of the lungs. So I should have a whiteboard by now, but I don't. So we're just going to do paper. Actually, we're not. We're going to wait till we get a diagram so I don't have to do anything. Identify the muscles used for quiet inspiration, forced during forced inspiration, and during forced expiration. I think we can just actually just cut ahead and I can answer that. So we already learned about our intercostal muscles. So the external intercostal muscles are going to be in the outside of the ribs, and the origin point is our spine. So here's our ribs, and here's our spine. My thumbs would be like the spine. If it's going to pull, it's going to pull it away. It's going to actually help us breathe. So whenever we try to think of deep breaths and people say to open up your chest, that's basically what you're using, your external intercostal muscles. and your internals are just going to be on the inner lining of those ribs. So if they're going to be on the inside of them, they're going to help to pull inside. So that's when we're and that's the difference between quiet respiration when we're just kind of normally breathing versus when we're using energy to expire. So, and also explain how the following affect pulmonary ventilation: bronchial smooth muscle contractions. And another way we can think of how bronchial smooth muscle contractions affect breathing is asthmatics who have constricted and thin, stronger linings of bronchial smooth muscles that don't help them breathe. Lung and thoracic wall compliance, recoil, and the pulmonary surfactant and alveolar surface tension. Um, so, if compliance is how stretchy your lungs are, how willing they are to move. So, if they're not willing to move a lot, then you're not gonna like. It's like the difference between pulling an accordion all the way or versus a little. Like it's just you're just gonna get more out of being able to stretch more. And recoil is it's the lung's ability to squish out. So the recoil aspect is when it's getting extended. When it recoils is when it's returning back and expelling air. The pulmonary surfactant is used in the alveoli because they're two super small little muscles, small little balls. I mean, think of like bubbles. If the pulmonary surfactant makes sure that in any case that it just doesn't collapse. Because once it becomes so small, even water can and water is in there at all, the water will uh, shut it down. And let me think of a good example of water like that. We have already learned how water likes water, and so if you imagine if you put two wet pieces of paper together, once they touch even a little bit, they're going to be stuck together. So that's why we produce pulmonary surfactant, which lowers surface tension and prevents that. Alveolar surface tension is oh, just that I just described that. Describe how respiratory rate and ventilations are regulated. So describe major chemicals and neural stimuli. I don't think we've really explored this quite yet, but let's check it out. All right. So what does the respiratory do to you? What is the function of the respiratory system? Ventilate and perfuse gas, acid base balance. Why do we need a respiratory system? Yeah, I think right now I'm gonna switch to my notes, and then after we do run through my notes, we're gonna look at the slides just because it'll be, I think, a lot quicker that way. Because my slides are what I'm picking up from Sam on what we actually need to know. So pulmonary functions. There's a lot of typos in here, but I'll. So if you guys are trying to read, I'll go over it. I know my typo lingo. So part one, anatomy and physiology of breathing. So the functions are, of course, getting gas and exchanging gas. So ventilation or perfusion. Perfusion is another word for ventilation. Let's look at that. Perfusion, another word for ventilation. Okay, so perfusion is the process of blood flow to the tissues. So that's a good difference here. Perfusion refers to the flow of blood through the capillaries surrounding the alveoli, allowing the exchange the oxygen and carbon. So perfusion is at the alveolar site where there's gas exchange. Okay, so that's a good vocab word. So functions, of course, straining like coughing, speech, and acid-base balance, which we won't cover until week 5. Why do we need it? It's a gas exchange, and we've already learned that we need oxygen as the final electron acceptor for ATP in the electron transport chain. What are the respiratory gases? So we have there's a lot of other gases in the air we breathe, But the functional ones for us are, well, I shouldn't say functional. We'll get into that later. But the ones that we actually sort of deal with is oxygen and carbon dioxide. So molecular mechanisms, how does it do this? It goes from high concentration to low concentration, which is just diffusion. And so here are the four processes of respiration. We have pulmonary ventilation, which is inspiration and expiration. Pulmonary ventilation, again, it's just getting air in and air out. External respiration, external environment, is diffusion of oxygen and carbon dioxide down their concentration gradient from the um, environment, like the air in our alveoli, into the blood. Internal respiration, internal environment, diffusion of oxygen and carbon dioxide from the blood into tissue cells. So, yeah. So here I have only three processes. So let's check out the notes. Oh, three is transport of gases in blood. Whatever. That's whatever. We know that. All right. So major structures, we have conducting zones which only move air. So this is nasal cavity, oral cavity, pharynx, larynx, trachea, bronchial tree. And the respiratory zones are gas exchange, so surface within lungs. So here we got nasal cavity. That's just your nasal cavity. We know enough now to just kind of breeze past this part. Oral cavity, larynx. Larynx is above your trachea, being uh, above your trachea with the epiglottis at an open position, because we're not always eating. So it'd be a lot more beneficial to have the epiglottis open at rest. And of course, when we are eating, we close it. We close our epiglottis so that nothing gets into this. So here we go. Food's going in here. It's gonna fall down here. When this is closed, this is the epiglottis. This is gonna close itself over this little area. And then here is our esophagus. Well, this diagram doesn't really show the esophagus, but the esophagus line goes right here. All right, so bronchial tree. So even the bronchioles here, kind of deep into the lungs, they don't conduct. They're not. I mean, they don't exchange gas with capillaries, so they are not. They're part of the conducting zone. So respiratory zones are gas exchange, is where a gas exchange happens. So alveoli and respiratory bronchioles. And just because there was some confusion, I'll go over it again. We go from trachea. I'm going to start from the trachea. I'm not going to bother with this. Trachea to primary bronchus to, okay, sorry. Trachea, primary bronchus, secondary bronchus, tertiary bronchus, bronchiole, terminal bronchiole, respiratory bronchiole, alveolar duct, alveolar pore, alveoli. Okay, and that's good. That's 100%. All right, so let's go. Oh, okay. So this is pretty interesting. This is something that sucks because that we need to know, but because it's a little confusing. So the conducting zones are lined with pseudo. This is conducting. So this is just air. Are not. I mean, not just air. This is the conducting zones. We only are pushing around air. We're not diffusing air through these cells. So because of that, these are lined by pseudo-stratified ciliated columnar epithelium. Pseudo-stratified, just as a reminder, means that it looks like it's stratified. It looks like there's a lot of that these columnar epithelium are stacked on top of each other, but it's not. And ciliated means that they're cilia, which are kind of little hairs that dance around to move around mucus. And you might think, why is there mucus? We just the mucus is used just to clean out the air and trap particles and pathogens. And columnar, that's just the shape, and epithelial, that's just the tissue type. So with goblet cells, oh yeah, and goblet cells. Goblet cells are, as we learned in Anatomy and Physiology 1, produce mucus. So this keeps the air moist, which we need. We need it to be moist. Air inside of our lungs is at 100% humidity, so it needs to be very moist, and it traps particles inhaled. Cilia move the mucus up and out like a mucus escalator, whatever that means. All right. Respiratory connecting zones lined by pseudostratified epithelial. Uh, Goblet cells produce mucus. Cilia. Okay. All right. So here we got today. We got a. I'm filming this on Monday, April 7th, our third class. So today we got a quiz about this. So let's just go over it again. So the frontal sinus, I'm going to start on the right hand side. The frontal sinus is pretty easy to remember just because it's at the very front and it's a sinus. The ethmoid bone, or your hard plate, is the ethmoid bone the hard plate? Oh, whoops, I'm looking at the wrong area. The ethmoid bone is above your nasal cavity. And here's the olfactory epithelium. That's what's breathing. For smelling, nasal conchae act as turbines, uh, and it just kind of makes it spins around the air so that more mucus it is more likely to get pushed into mucus, and so that we are more likely to effectively clean the air we breathe. Nasal vestibule is just the entrance. Nostril, hard plates, soft plates, soft palate. Sorry, I meant to say palate. Tongue, lingual tonsil. That makes sense because it's right behind your tongue. And palatine tonsil on the other side here. I'm skipping. Makes sense because it's next to your soft palate here. Uvula, epiglottis, hyoid bone, thyroid cartilage, cricoid cartilage, vocal fold, vestibular fold, laryngopharynx, oropharynx, fauces. Okay, so that's the opening here. That's good to know. Uvula, nasopharynx, opening of auditory tube. Okay. Nasal metasis. Huh. I guess we'll learn the practical what we actually need to know. So conducting zone, we got the nasal cavity. Nasal cavity structures work together to warm air, humidify, and filter air. So this has respiratory epithelium. So this has that ciliated columnar, pseudostratified ciliated columnar epithelium. So the mucus traps debris and pathogens with nasal hairs, and it filters out large particles like with the use of nasal conchae. So overall, this is just used to warm up the air. The nasal conchae could also be something that warms up the air. Like there's just so much more surface area and so much more meat to warm up the air we're breathing. Force air into turbulence to increase chances of all the air will encounter the epithelium or mucus. And we also have paranasal sinuses, uh, which secrete mucus to also warm and humidify. So that's the overall goal of that. All right, so here we're going to go into some more anatomy. The pharynx is a tube formed by the skeletal muscles and lined by a mucous membrane continuous with the nasal cavity divided into 3 zones. So a tube formed by a skeletal muscle and lined by the mucous membrane continuous. When it says mucous membrane, it's basically saying that it's part of that pseudostratified ciliated columnar epithelium. And so it's divided into 3 zones. So the pharynx isn't a specific part. It's this whole area here. And those 3 zones are the nasopharynx. So here we can see the colors. The nasopharynx is the part of the pharynx. So here I'm going to circle around what is the pharynx. Basically, what is colored. The upper portion by your nose at the nose level, as we can see here, is the nasopharynx. Uh, and then by the mouth, it's the oropharynx, and then right here is the laryngopharynx, which makes sense because this is the larynx, so larynx, pharynx. So you kind of see how it's the meeting point between the pharynx and the larynx is the laryngopharynx. If that, sorry if I'm being confusing here, but it should make sense if you're watching, so you can just look at what I'm talking about. So nasopharynx, passageway for air only. So yeah, that's another thing to think about is where food is going to touch. Food is going to be touching the laryngopharynx and the oropharynx, but not the nasopharynx. So it's for air only. Tonsils trap and destroys pathogen. Soft palate and uvula block passage to the nasal cavity while swallowing. Oropharynx, passageway for air and food and tonsils. All right, so here's the conducting zones are normally pseudo stratified columnar epithelium. However, we have to consider where food is touching because while we're eating food, it's a lot. We eat things that are sharp, so we gotta like not have that more sensitive epithelium. We need stratified squamous epithelium for where food's going to touch in order for us to protect the lining of our mouths, which is obviously really important to eat and metabolism. It's basically, yeah, we need to protect the oropharynx and the laryngopharynx because the pseudostratified ciliated columnar epithelium isn't going to be enough. We need to have that strong, very strong stratified squamous epithelium. Air and food tonsils destroy pathogens, passageway for air and food, ends as larynx and esophagus split, lined with stratified squamous epithelium. All right, so now we're on to the larynx. So the larynx connects the pharynx and the trachea. Yeah. So it helps regulate the volume of air entering and leaving the lungs. Epithelial transitions. So so it transitions from stratified squamous epithelium to pseudostratified ciliated columnar with the goblet cells, which makes sense because here, because once we get into the larynx, food is not going to be touching it anymore. This area, when there's food, this on my cursor, the epiglottis goes down to cover itself, which is cute. So major structures, thyroid cartilage. Oh, okay. So this the thyroid cartilage is this big kind of frontal cartilage piece here. Hopefully we get a better photo, but it's big, and we saw it from the assignment we're doing and from the dissection. And the cricoid cartilage in all these photos is this part. Here you can see my cursor. These two lines, it kind of forms like a ring around it, and it's kind of tilted up here. So this and the rest, you can kind of see it's its own like self-repeating pattern, and that's just tracheal cartilage. And so the epiglottis here, we're entrance and guard to the larynx. When swallowing, epiglottis covers glottis, food directed. And then once this closes, here you can imagine this flap falling over here. Food is just going to go right down the esophagus. And so this vocal fold here, this line that we see, is a sound. It's used in sound production as air vibrates vocal folds, and false vocal folds protect the vocal folds. Let's see my notes about that. Just some more. I'll finish this slide. Oh, I did. Okay. And we're just gonna catch up to that point on my notes. Okay, so paranasal sinus, pharynx, we know nasopharynx, we know larynx, we know. Yep, major structures. So vocal folds, sound production, air vibrates vocal folds. Okay. Looks like I have no more notes on that. Let's just look it up. False vocal cords versus true vocal cords. So the false vocal cords, vestibular folds instead of vocal folds, are located above the true vocal folds, uh, vocal cords in the larynx. And while they play a role in air protection, they are primarily involved in sound production like the true vocal cords. So it looks like we got some photos here we can look at. True vocal cord, false vocal cord. So this is a top view. You're looking down. Um, so it looks like they close to protect our vocal cords down there. I don't know enough to identify that. All right, so vestibular fold is basically above the vocal cord, and it's basically just protecting. And again, I don't know how much, to what extent we need to know for these following exams. All right, so now we're going to the. I guess we can switch back to the slide just because there's photos you guys can refresh yourselves on. Right, so tracheal cartilage again. Cricoid cartilage is this looping back, this ring here that kind of gets chubbier on the backside, and the thyroid cartilage is this front kind of bold area here. All right, so the trachea is a tube supported by C-shaped rings of cartilage. Why is it C-shaped? It's because the esophagus is here. Here you can see this photo. It's like, why wouldn't you just make the full thing, do the full whole dang thing? And it's because it's kind of sharing some structure in the wall with the esophagus. So that's why if you get something stuck in your throat while you're eating, this thing would push into the lumen of your trachea, and that's why you can't breathe. Tube supported by 15 to 20 C-shaped rings of cartilage is always open, pseudostratified ciliated columnar epithelial with goblet cells. Again, how do we know that this is going to have pseudostratified ciliated columnar epithelium with goblet cells? It's because it is a conducting zone and it doesn't deal with food. Those are our two marks. So bronchial tree. Bronchial tree, mucous membrane that traps debris and pathogens and branch reach to alveoli. So the bronchial tree is part of the conducting zone because it's not exchanging air with capillaries. So we go here to the primary bronchus. Once we branch off, this is the secondary bronchus. This is the secondary bronchus. If you guys can see my cursor, 2, 2, 1.